Hello, my name is Miller Devane, and this is my Journal Club presentation. So the article I chose was the analysis of statistical and standard algorithms for detecting muscle onset with surface EMG. The objective of this article was to evaluate 6 different classes of standard and statistical algorithms to determine muscle onset in both experimental and surface EMG with a simulated EMG, and a simulated EMG with a known onset time. So the subjects were 18 participants, which were 13 males and 5 females that had no previous surgeries, current or recurring pain, or injury on their dominant side. For the inclusion-exclusion criteria, they didn't specify it any further than saying that the inclusion criteria for participation was the absence of neurologic, cardiovascular, or metabolic disorders. For the representativeness of the sample, there was a gender imbalance and a limited age generalizability because the age range they chose was pretty short. So people on the really high and really low end of that age spectrum wouldn't really necessarily be able to generalize these results. And all of the participants were right side dominant. This excludes populations with pre-existing conditions, and it is also not fully representative of populations with diverse health conditions, handedness, age groups, and different muscle composition. For the for the ethical considerations, this study was approved by the U.S. Army Research Lab Institution Review Board, and participants gave their written informed consent in accordance with the Helsinki Declaration. For the method and design, there were 18 adults, as mentioned, and the participants performed 3 repetitions of each movement, elbow flexation and knee extension, with a 2.5 or 2.3 kilogram weight. The EMG data so the electrodes for the EMG data were placed on specific muscle locations following skin preparation to reduce impedance and noise. These signals were then amplified, bandpass filtered, and sampled at 248 hertz. The visual detection, researchers performed double-blind visual EMG onset detection twice for each trial, with results averaged to establish a gold standard for comparison. For the simulated data, the experimental data was altered to simulate known muscle onsets, ensuring objective assessment of algorithm accuracy. For the ethical considerations, this study adhered to the Declaration of Helsinki and ensured respect for participant autonomy, beneficence, and confidentiality. The participants provided informed consent, as mentioned before, and was also reviewed and approved by the U.S. Army Research Lab. So the data was analyzed using a multi-step approach. It was split into the different algorithm classes. So there was the standard methods, which was linear envelope, TKEO, and sample entropy. And then the statistical methods, which is general time series analysis, sequential and batch change point detection, and Bayesian change point detection. For the analysis steps, the visual onset comparison, experimental EMG onset detected by algorithms was compared to the gold standard visual detection, which was that average that we talked about earlier. The simulated data comparison was that algorithms were tested on simulated EMG data with a known onset. For the performance metrics, root mean squared error was used to compare algorithmic EMG onset detection to the gold standard or known onset, while linear regression was used to determine the effect of signal to noise ratio on algorithm accuracy, and with algorithms sensitive to SNR excluded. The mean differences and confidence intervals. So the remaining algorithms that were assessed were evaluated for their average error, bias, or reliability, which is their narrowness of their confidence intervals. For the Bayesian analysis, posterior probabilities and hyperparameters in Bayesian change point analysis were systematically varied to optimize performance. As for the appropriateness of these statistical tests, linear regression helped identify algorithms biased by SNR, which ensured reliability across the varying EMG signals, while root mean squared effectively measured the accuracy of algorithmic onset detection compared to the benchmark values. The use of confidence intervals provided insight into both accuracy and reliability, while the Bayesian change point analysis, with its probabilistic framework, was particularly well suited for detecting EMG onset in noisy and variable conditions. The key results they found were that the Bayesian change point analysis outperformed the other methods in both reliability and accuracy. The standard methods had reasonable results, but their wider confidence intervals reduced the reliability of their results. The Bayesian approach showed a superior performance even in low quality or noisy EMG signals. For the statistical significance, algorithms with statistically significant performance were retained during regression-based down selection process. Bayesian change point algorithms had mean differences close to 0 with confidence intervals that overlapped the gold standard or true onset. For the findings of the study, the Bayesian change point analysis, it shows the posterior probability right there. was clearly the most accurate and reliable method for detecting EMG onset, while standard methods like TKEO and linear envelope were less reliable due to their wider confidence intervals. Bayesian methods performed well on both experimental and simulated EMG data, even with noise present. For the presentation and interpretation, results were presented clearly using figures like the forest plots shown to the right and shown before to show mean differences and confidence intervals. Key algorithms were compared effectively and efficiently, while the Bayesian change-point analysis is superior for detecting EMG onset, especially when signals are noisy and have variable quality. The authors emphasized the method's robustness and suggested it as a standard for future studies. As for the limitations, like we talked about earlier, there was a small homogeneous sample, majority male and all right side dominant. It excluded individuals with neurological or musculoskeletal conditions, which limits clinical applicability to people with pre-existing conditions like that. It was focused only on single onset detection, not multiple bursts of muscle activity, which opens the door for a similar study to be used on those multiple burst muscle activity detection. This is kind of what we discussed earlier with implications of the study. It provides a more reliable and more accurate method for detecting EMG onset, which is a standard approach for analyzing noisy or variable EMG signals. As for contribution to knowledge, it introduces advanced statistical techniques from other fields into biomechanics research. And it demonstrates the limitations of traditional methods like TKEO and the potential for Bayesian methods to improve accuracy and reliability. As for replication feasibility, the study is replicable and access to appropriate EMG recording tools and the statistical software to calculate those. So openly available raw EMG data is on GitHub and ensures transparency and reproducibility. And they do link their GitHub at the bottom of the article. The additional questions that were raised by this article, or I guess you could assume are raised by this article, are how effective is Bayesian change-point analysis for detecting multiple muscle onsets during complex dynamic movements? And can the methodology be adapted for clinical populations, example, individuals with neurological or musculoskeletal conditions? Another question is, how would varying the Bayesian hyperparameters further optimize the method across different scenarios? And that's all. Thanks.